Today, myself and my colleagues are going to be talking about xenotransplantation of organs generated through blastocyst complementation. My name is Melanie Rojas, and my associates are Zachary Lawrence, Varun Koda, and Roberto Filani.
Thank you, Melanie. Now, I'm going to explain how this has any conjunction with diabetes. The facts being 37.5 million Americans have diabetes, but 1 in 5 don't even know about it. And we've seen an ever-growing trend in the prevalence of diagnosed diabetes amongst adults aged 20 years and older. Furthermore, type 1 diabetes especially impacts the pancreas, and it's imperative to develop pancreatic organs that benefit these individuals. Despite this, we've seen over 100,000 people on the national transplant waiting list, as 17 people die each day, and every 10 minutes in a day, another person's added to this waiting list. It's clear that there are glaring flaws regarding the organ transplantation process, as the team and I have identified limitations of this entire procedure, since there is a shortage of available organs to transplant into recipients. Furthermore, if a recipient were to receive an organ, there is an issue oftentimes of properly matching an organ to the recipient, as an improper match can result in organ failure. This is critical since a pancreas transplant works to reduce the impact of type 1 diabetes. However, an improper organ or an improper match can result in death. As a result, my team and I are working and have developed a proposed method to resolve this issue, and we like to do so with our thesis, as we'd like to focus on applying research towards the pancreatic cells within pig and mice to xenotransplant these cells into humans to recreate organs for those requiring organ donations. Typically, other animal organs are rejected by the body due to an immune response, whereas we'd like to utilize Blastocyst complementation in conjunction with iPSCs, as we aim to alter an animal's organs to respond positively with a human's immune response system. This idea is based off of the fact that we currently use pigskin to treat burns, as well as other animals to medically benefit humans. With all of that in mind, it's imperative to define some key terms, the first being xenotransplantation. It requires the transplantation of organs from a non-human or animal source into a human recipient. We also plan to leverage induced pluripotent stem cells, or iPSCs, where they are immature cells generated from an adult mature cell and have the ability to become any type of cell in the body. Furthermore, we plan to leverage and utilize blastocyst complementation, the knocking out of genes in an embryo to allow for the injection of donor pluripotent stem cells into the host blastocyst. Now, my colleague, Ravi, will explain more about our proposed methodology.
Thank you, Varun. So as Varun explained, the goal of this research is to essentially grow human pancreas in pigs. Now, how are we going to do this? Well, the first step is to first knock out the genes that create the pancreas in pig embryos. The gene that is responsible for the differentiation of the pancreas is the PDX1 gene. So we're going to be removing this gene with CRISPR-Cas9 technology, essentially generating embryos of pigs without pancreas. Then we're going to be injecting induced pluripotent stem cells into these pancreasless embryos, essentially replacing the pancreas of these pig embryos with human cells. So as the embryo develops, it will develop into a human pancreas. Now, where are we going to get these stem cells from? Well, these stem cells are induced pluripotent stem cells, which means that they're derived from adult human cells. You can simply take the skin cells of an adult donor, treat them with some transcription factors, and then essentially deprogram those mature cells into immature stem cells that can be injected into these pig embryos to produce pancreas. It's a very neat method because it escapes the ethical considerations and red tape involved in embryonic stem cell usage. From there, we will let these pig embryos develop and identify their pancreas. See if the manipulation worked. Do the pancreas successfully incorporate human DNA and human stem cells into the pancreas? So we're going to be running some chimeric assays to essentially test to see if human pancreas really did grow in these pigs. If they did, then, and if this research continues to be funded, we can move on to clinical trials, taking these human pancreas from pigs and directly transplanting them into diabetic human patients and seeing if these new pancreas increases insulin levels to treat symptoms of diabetes. So here are some more visuals for how this process is going to work. We're going to use a transgene to knock out pancreas-producing cells in the blastocyst of swine. Then we're going to be injecting induced pluripotent stem cells from the donor of a human, essentially, as the pig develops, producing human pancreas in a pig. And after we analyze the pancreas for its human content, we can transplant that into a human subject, and with the help of immunosuppression, essentially generate a functional pancreas that will treat diabetes. So just to reiterate, again, we have these somatic cells that are deprogrammed into stem cells, injected into an embryo that is lacking a pancreas to produce a chimeric pig with pancreas of a human, and then injecting that human pancreas back into a human diabetic patient.
Next, we kind of dive into the ethical considerations of our research proposal, and this also taps into kind of what are the standards that we are upholding ourselves during this research program. So some of the policies that we looked into were FDA guidelines for xenotransplantation from animals to humans, NIH guidelines for animal testing, IRB approved protocol for future testing involving human subjects. We also included city training, which will be completed by each researcher on the team before any work is done to ensure that everyone kind of knows what they're doing and is responsible with what they're doing. And we also included ICOOK guidelines and protocols for animal testing to make sure that no animals or future humans are being harmed in any of these procedures. Potential funds that we could expect for our research kind of dives into these four main categories. The American Cancer Society and the American Diabetes Association being two of the biggest ones, because our research does have some pull in pancreatic cancer specifically, and also type 1 diabetes. So that falls under those two categories, with both of these societies or organizations having grants and funds specifically for pancreatic cancer or type 1 diabetes, but we can also apply to more generalized grants as well. We can also apply to some of the institutions that are within the NIH, because again, we have those two major ties with cancer and diabetes.
So what can we take away from this research? Essentially, before we go into that, I would like to discuss the trends that this research was based off of. So this experiment builds off a previous experiment that was able to successfully xenotransplant liver cells into pigs and mice. So we saw that these researchers were successful in this xenotransplantation of Blastocyst Complementation derived organs. And previous work has also shown to improve chimeric efficiency of cells, tissues, and organs produced by non-human species, so that the organs that we are producing are able to transplant into human subjects, because those are human cells that are being developed in those non-human species. So this study aims to prove that blastocyst complementation organ generation in conjunction with xenotransplantation can be used to generate functional pancreatic cells in pigs and mice that are producing and secreting insulin to allow the body to take up and utilize glucose. We also aim to optimize this method to increase the effectiveness in higher level organisms to eventually use this method in humans. So we wholeheartedly believe that this method can be used to treat terminally ill patients suffering from refractory diseases. As previously mentioned by Varun, these statistics are horrifying, and in most cases, these are ideally treated with an organ transplant operation. However, there is a shortage. If follow-up experiments are approved, we aim to utilize this method to generate personalized organs for these patients, therefore decreasing the problem with the shortage of organs and increasing the compatibility between organ and donor, because we are utilizing the patient's own cells. This specific method will aid patients suffering from terminal pancreatic cancer and type 1 diabetes.
Thank you for listening. And this is our team. you have any other questions, please don't hesitate to reach out to us.